Hello students, I am Dr. Vinod Shinde. In the previous lecture, we have discussed about the tricarboxylic acid cycle or TCA cycle, also named as Krebs cycle, which is oxidative metabolism of pyruvate or pyruvic acid and pyruvic acid oxidation in the presence of oxygen in the aerobic condition. The metabolism of pyruvic acid occurs and it generates 30 ATPs. Now we will discuss about the respiratory electron transport system or electron transport system. In eukaryotes, the electron transport chain and the enzyme of Krebs cycle are found in the mitochondria. The Krebs cycle enzymes are located in the matrix of mitochondria, while the components of electron transport chain are located in the inner membrane. A number of dehydrogenation reaction takes place during the cell respiration, one during the aerobic glycolysis or EMP pathway, one during the oxidation of pyruvic acid, that is TCA cycle, and four during the one during the oxidation of pyruvic acid, that is pyruvic acid to acetyl coenzyme A formation, and four during the Krebs cycle generation or Krebs cycle turns. That is the hydrogen that we have discussed. The four, one during the glycolysis occurring is in the formation of three glyceraldehyde acid, glyceraldehyde 3 phosphate to 1 3 diphosphoglycerate, and here we have one RETC respiratory electron transport system occurring. Then second is the formation of acetyl CoA from pyruvate, that is pyruvate oxidation. Here NAD to NADS2, here we have RETC occurring. Then in the reaction of isocitrate to oxalosuccinate, we have RETC occurring, that is NADS2, and alpha ketoglutarate to succinyl CoA. Here also NADS2 is occurring. Then we have succinate to fumarate. In these, two hydrogen molecules are releasing, that is FAD to FADS2. This is also a RETC, respiratory electron transport system. Then malate to oxaloacetate. Here also RETC is occurring with the generation of total 38 ATP molecules. The two hydrogen acceptors in all but one of these reaction is nicotinamide adenine nucleotide, that is dinucleotide, that is NAD. Nicotinamide adenine dinucleotide is NAD in all these reactions, but one is FAD. The hydrogen released during the reaction of succinate to fumarate is FAD, that is flavin adenine dinucleotide in the Krebs cycle. The reoxidation of NAD FAD takes place by the passage of electron through a series of intermediate carriers. These are flavin mononucleotide, that is FMN, and of flavoprotein coenzyme Q and ubiquinone and cytochrome BKBT. As the electron passes down the chain, there is alternate reduction and oxidation of its components. The carrier are arranged in the series of gradually increasing redox potential. Electron tend to pass from the carrier with a more negative redox potential to the carrier with a more positive redox potential. For each pair of electrons passing down the electron transport system, 3 molecules of ATP are generated, one at each of the 3 sites of the chain. These are the site of oxidative phosphorylation. In the site 1, between NAD and coenzyme Q, we have a production of ATP, that is 3 molecules of ATP. In the site 2, between cytochrome B and cytochrome C, we have ATP generation, and in the site C3, that is between cytochrome C and oxygen. The final hydrogen acceptor is molecular oxygen, and it is because of this that most of the organisms require oxygen in order to survive, as RETC produces ATP molecules with the uh, acceptance of hydrogen molecules or hydrogen ions released during the Krebs cycle or respiratory electron transport system. The entire respiratory chain and the three sites of ATP generation are as these. Here, NAD and FAD, two molecules, work more. In that also, NAD is more dominant than that of FAD. We have all the reactions converting NAD into NADH2, and only one reaction, that is succinate to fumarate. Here we have FAD molecule. NAD, NADH2, NADH, that occurs simultaneously, giving you a simultaneously giving you the ATP generation in the three sites, that is coenzyme Q, then coenzyme Q, cytochrome C, and cytochrome B. The ATP generation is the main issue in this respiratory electron transport system. In each site, we have three ATP molecules generated from one NAD molecule. So, one NAD molecule, we have three ATP molecules, and we are working in the Krebs cycle or glycolysis with the two molecules. So, it will double the production of ATP. So, this is the respiratory electron transport system in the eukaryotes. In the prokaryotes, the respiratory electron transport system generates ATP in a similar way, giving you a more ATP generation in the system. Here important is NAD, NADH2 formation, FAD, FADH2 formation, nicotinamide adenine dinucleotide, and flavin adenine dinucleotide. These two molecules are important. Of these, abundant is NAD, that is nicotinamide adenine dinucleotide molecule. Now, this is the ATP generation in the respiratory electron transport system. In the next lecture, we will discuss about the metabolic mill, or the interrelation between EMP, HMP and ED pathway, and energy generated during these cycles. Thank you.